Dr. Burns is a plant ecologist at Case Western Reserve University, and research in the Burns lab addresses questions about plant interactions, such as what makes some species invasive, how do plant interactions below ground influence resilience to stressors, such as disease and climate change. And today, Dr. Burns is going to be speaking about rhododendron pests and diseases in a changing climate. So take it away. I will stop sharing and please take it away, Dr. Burns.
All right, thank you so much, Juliana. I'm gonna go ahead and share my screen. Please, someone like Juliana, holler at me if you can't see my screen. Let me know if you can hear me okay. Seem fine, awesome. Okay, so thank you guys so much for joining us today. I'm really thrilled to share with you a little bit of some of our recent research on rhododendron and why I am falling in love with them as a study system. As I mentioned, I'm a plant ecologist, and so my background is in understanding basic plant ecology and how plants interact with their environment and the world around them. And plant ecology, like many disciplines, has some powerful abilities to address pressing global problems. And one of the global problems that we are particularly pressed to address right now are costs to our food security. You may already know that 10 to 40% of crops around the world are lost to pests and diseases every year. So plant pests, although they might seem like an obscure academic interest to some degree, also are very practical to understand. In a really dramatic example, the pathogen Phytophthora infestans causes the disease potato late blight. And you can see on the left here an image of a potato that has been damaged by this pathogen, and you can see that they are entirely inedible. This pathogen is native to South America, and when it was introduced in the 1800s to Europe, it caused the Irish potato famine. Over 1 million people died as a result of this single pathogen introduction, and 1 million more were displaced or migrated to other parts of the world. And the pathogen persists to this day. It costs around $6 billion a year globally, so it is terribly damaging. And these kind of agricultural costs of plant diseases are not the only potential problems caused by plant diseases. There are also ecological costs as well that occur as a result of many plant diseases. For example, the closely related Phytophthora cinnamomi causes the plant disease sudden oak death, amongst other diseases. And when a plant is infected with Phytophthora cinnamomi, you see here a plant that has been infected and is rapidly dying. Again, this is an invasive plant pathogen that was introduced outside its native range. Here you see it in its introduced range in France. This is its distribution in France historically. And historically, the distribution of Phytophthora cinnamomi has been limited by cold winters. So cold winters knock the pathogen back and reduce the severity of disease. So you see here mapped across a map of France, the severity of sudden oak death disease, where red colors are more severe disease and up in blue is essentially no mortality from disease. So what you see is that historically, the range of sudden oak death was limited in France by cold winters, where most of the severe disease happened in the south and west of France. But that is projected to change with changing climates. And as winters get warmer, we are projected to see an increase in the distribution and severity of diseases like sudden oak death. So that is to say, climate change is making some of these problems, like invasive plant diseases, worse over time. And how can we address this pressing problem of changing climates and diseases interacting to influence plant health? Well, I really want to emphasize that this is a multidisciplinary and a multi-scale problem. So I'm certainly not going to claim that we're going to address all of those problems in one study system or in one talk today, but I just want to emphasize the opportunity there to do interdisciplinary work and to do work at multiple scales. So you may already know that climate change affects plants and disease interactions at many scales, from the microclimate scale to the macro scale. And there's very strong evidence at the micro scale that for example, plant physiology responds dramatically to environment and to changing climates. For example, many plants are very good at closing their stomata, the pores on their leaves, when they are exposed to drought. And so with increasing drought severity, we can expect that some plants will respond in their physiology in a very adaptive fashion and will be drought tolerant, whereas other plant species may be less drought tolerant. So understanding the basic physiology of plant responses to drought is essential for understanding the effects of shifting climate. But similarly, at larger spatial scales, like the scale we saw in France for Phytophthora cinnamoni, there are regional scales at which shifting climates will change the distribution, abundance of species, and even the severity of disease. So essentially, this problem of how will shifting climates affect diseases, how will that interact or scale up to affect plant health, is an interdisciplinary and a multi-scale problem. And one of the things that I hope to convince you of today, and one of the things that I'm becoming increasingly convinced of, is that rhododendrons are a really excellent study system for addressing some of these pressing problems. And one of the things that I think is really valuable about them is that they have this interesting intersection of ecology and evolution. So one of the things I'm going to tell you about today is this disease, root rot. We call the disease root rot in rhododendrons. It's caused by Phytophthora cinnamoni, which I already mentioned earlier. Phytophthora cinnamoni is a pathogen that is an oomycete. That is, it is a water mold. It used to be classified as a fungus. It's no longer classified as a fungus. But essentially, it is a pathogen. It's waterborne. It has waterborne zoospores, so it spreads under moist conditions. And when you inoculate rhododendron plants that are vulnerable with this pathogen, you see that they show symptoms of disease that are sometimes very dramatic. They often die very quickly. And on the right, these are plants that are under control conditions that haven't been inoculated with the pathogen. So from an ecological perspective, that's very interesting because there is this known pathogen that, as I said, it can be inoculated experimentally. So it has some nice experimental properties. There are species that are quite vulnerable to the disease. As you see here, this rhododendron minus species is very vulnerable. You can see that it's dying here. But interestingly, from the sort of evolutionary perspective, disease susceptibility occurs multiple times across groups of rhododendrons. So this leads to replication, which I'm sure you all know, scientists are very fond of replication. Basically, anything where there is replication across groups gives us more power to address general questions. And that is especially the case in rhododendrons for susceptibility to Phytophthora. So I'm going to tell you quite a bit more about this data, but just to give you a sneak peek of it now, there are 4 clades in the rhododendron that we have sampled, including the deciduous azaleas, the Phytophthora, the Sutsutsi, ponticum, and rhododendrons. And within each of those groups, there is variation in disease susceptibility to this pathogen. So I've marked those species that are very susceptible to the disease with a bright pink here. That is, if you add the pathogen, they die. Sort of like rhododendron minus is in this picture. But its close relative, Rhododendron kieskii, really does not die at a very rapid rate when you add the pathogen. It's much less susceptible to the disease. And you can see that across groups, there's variation in how susceptible these species are. So we had a hint of this from the literature before doing some of our experiments, but I'm going to show you what I think is more convincing experimental evidence today that there is replication of disease susceptibility across these groups. And that's very powerful for addressing general questions and pressing global problems like how are shifting climates going to influence the severity of disease. So I mentioned Phytophthora cinnamoni several times. It is this, I said it's a water mold, it's an oomycete pathogen. It causes the disease root rot in rhododendron species. And you can see here, certainly the symptoms start in the roots, but you can see those symptoms above ground after a pretty short period of time. Once the plant's roots begin to rot, it can no longer take up water, it starts to wilt, the leaves begin to discolor, and in some cases, the plants die very rapidly. And this is not just a rhododendron problem. Phytophthora cinnamoni, I also mentioned that it causes sudden oak death It can affect over 5,000 species worldwide, including many woody species and including some crops like avocado. It costs somewhere upward of $40 million a year in avocado crop losses just in California every year. So this is potentially a very broadly important disease. And the Phytophthora pathogens in general, as I mentioned with potato late blight, they're sort of a general class of pathogens that affects a broad group of plants, and it is sort of very important globally. And as with many diseases, prevention is cheaper than cure. If you were to ask, for example, for cultural recommendations from the American Rhododendron Society, they would tell you all of the good classic things which do work, like plant in really well-drained soil, plant in a raised bed if you can, try to keep mulch away from the very base of the plant, that is mulch, but not up to the very trunk, plant high up, add organic matter, all of that good stuff. And anything that improves drainage does reduce the chances of root rot because, as I said, it's a waterborne zoospore that causes infection. So it is very helpful to improve drainage. But as you know, if you've been watching the news recently, the incidence of severe weather and severe flooding is increasing. And that's true both in moist areas of the world in panel A, all the way up to the most arid regions in the world in panel D. So you see here the change in precipitation or extreme flooding events projected over time, and the darkest blues are extreme flooding projections. So the incidence of extreme flooding is projected to increase even in some of the most arid regions in the world. So even my relatives in Arizona are experiencing not only historic droughts during some parts of the year, but also sometimes in the same year, historic flooding events. So that makes the problem of prevention more challenging going forward. And in conversations with nursery owners, they have told me that they still need more prevention tools for problems like root rot. So they need cost-effective protection that lasts. There are protection mechanisms. There are chemicals you can add like phosphite. It requires frequent reapplications. So even though it's relatively inexpensive to buy, over time it is not necessarily cost effective. And so nursery owners have told me that they frequently don't use it. So they really need prevention tools that are cost effective and that last. So it sounds like we're facing a conundrum, right? There's lots of kind of pressing problems that are intersecting here, from climate change to disease. How can ecology help us to address or start to address some of these pressing problems? I teach ecology, and I'm teaching ecology this fall. And in my ecology class, I like to teach my students about the idea of emergent properties. Emergent properties are a property of a whole community that emerge or come out of all of the ecological interactions, all of the predation, mutualism, parasitism, everything that happens in the context of a community might lead to, on the whole, some sort of larger property, like carbon sequestration, or in the case of a soil community, plant health. This larger property emerges from the community as a whole, and it may be difficult to predict from just knowing the component parts of the community. So just knowing a single player in the community might not be sufficient to project that emergent property. And in the example I give in my class, I give this example of a very kind of classic, simple marine system. It occurs off the coast of North America. And there is a primary predator in the system, sea otters, which are known to eat sea urchins. They eat a lot of sea urchins. And sea urchins, in turn, feed on kelp. Where there's a healthy population of sea otters, they keep the sea urchin population in check, and the result is a large kelp forest with lots of carbon sequestration. That's great from a climate change perspective, because we would like to keep systems intact that have that emergent property of a lot of carbon sequestration. On the other hand, on the right, you see this alternative scenario where the population of sea otters declines, perhaps as a result of hunting or other pressures. The result is a boom in their prey, the sea urchin, and the sea urchins eat kelp, resulting in a loss of that kelp forest and a reduction in carbon sequestration. So you can see how if you just knew a single component part of this system, you would not necessarily be able to predict what is the amount of carbon that is sequestered off the coast of North America. But if you understand all of the players as a whole, if you measure carbon sequestration as a whole, you can get at this emergent property of carbon sequestration. Well, what does this have to do with plants and their diseases? When we study plant communities or plant populations, we have found that there is a tremendous influence of the soil microbial community at the plant's feet on plant health and on plant interactions with disease. And this is a tremendous literature, but just to give you a kind of snippet of it, plants do a lot of active things that influence the microbes found in the soil. Plants are not passive responders to their environment, including to the microbes in their environment. In fact, they produce root exudates, they produce volatile organic compounds, for example, that are emitted by the root that improve pest tolerance in some cases, and theoretically can reduce the use of pesticides. They also produce organic acids, which are involved in improving nutrient availability and can reduce the dependence of plants on fertilizers. And plants have an incredibly complex microbial community at their feet: bacteria, fungi, oomycetes. The bacteria community in some cases includes rhizobia bacteria, which are well known to be mutualists, and mycorrhizal fungi, which are also mutualists in many cases. And on the right, you see just a small snippet, just a few species of many species of mycorrhizal fungi. These happen to be ectomycorrhizal fungi, which ensheath the outer part of the root. But rhododendrons, for example, also are famous for their relationships with ericoid mycorrhizal fungi. So the mycorrhizal community alone is incredibly diverse and full of many mutualists and even, under some conditions, pathogens. So this community is complicated, is sort of the short answer. But as a whole, the community of microbes has an influence on plant health that can be thought of as an emergent property of that soil microbial community. And ecologists have been studying that emergent property for many decades, in some cases using a whole soil microbial community experiment. And what many ecologists, including our lab, have done is they've taken live soil, so live soil microbial communities, perhaps conditioned by the plant, that is influenced by that plant. You can dig up the soil around that plant's root, and you can compare the effect of that live soil biotic community to a sterilized control, for example, autoclaving that soil. You add a small amount of the inoculum to a pot, and then you ask the plant, what is the emergent property of this soil microbial community on your health or biomass as a whole? And the result on average across meta-analyses, including some of our meta-analyses and some of Carrie Crawford's work, show that on the whole, most plants respond fairly positively to that soil biotic community. So you see kind of a larger plant here in the live treatment than in a sterilized treatment, because on average, most plants are benefiting from that soil microbial community. Even though it may contain many pathogens, it also contains many mutualists. And we wanted to ask the general question, does the soil microbial community influence resistance to the pathogen Phytophthora cinnamomi, to disease? There's a lot of precedence for this question or addressing this question in the agricultural literature. And we thought there might be a benefit of the whole soil microbial community. We thought that not only because of these meta-analyses that have been done in ecology, but also because of the agricultural literature. So for example, if you were to look at take-all disease in wheat, it is considerably less severe in soils that have been conditioned by many years of wheat monoculture. So that's what you see in this figure. This is years of monoculture. So on the right is crops that have been in monoculture for longer, and this is disease severity on the y-axis. So you can see when the wheat crop has been in monoculture for only a few years, there's a great deal of take-all disease, so high amounts of disease symptoms. But where wheat has been grown in monoculture for year after year, the microbial community starts to develop a disease suppressive effect, which has been dubbed in agriculture disease suppressive soils, beneficial soil microbes, essentially. And so this is well understood for take-all disease. And so we expected, based on this, that there might be a positive influence of the soil microbial community on disease resistance, including to Phytophthora cinnamomi. But we also thought, well, okay, that seems like a nice prediction, but it does seem very simple, and it certainly could go the other way around. So before we did the experiment, we thought it might also be possible that that soil microbial community could be detrimental. There's also precedence for this in the agricultural literature. So if you've ever grown potatoes in your yard, you probably know that if you grow potatoes in the same patch of soil year after year, there is often a buildup of pathogens and more disease symptoms over time. So to successfully grow potatoes, as my parents do in Idaho in their backyard, they rotate the crop, right? They rotate the potato with other crops, especially other crops that are distantly related. So not in the Solanaceae plant family, not tomatoes or eggplant, something else. If you rotate with soybeans or broccoli or really anything that's not a Solanaceae, you're much better off. And that reduces the incidence of disease. So we thought that our whole soil microbial community, certainly it could potentially reduce the effects of disease, but it also could actually make the effects of disease worse by carrying in other pathogens and impacting plant health in that way. And I mentioned earlier that this study system is really, I think, kind of powerful for addressing these sorts of questions, in part because the pathogen Phytophthora cinnamomi is fairly well understood, and it can be cultured. So it can be cultured in the lab in vials. It can be stored in little vials, grown on hemp seeds for a while. And you can put it on an agar plate made with lima bean agar, and then you transfer the lima bean agar into like a rice grain. So I love the grant proposals that I'm writing with these methods, where I'm saying, oh yeah, I'm going to go to the grocery store, I'm going to buy some lima beans and some rice, and you use those things to culture Phytophthora cinnamomi. So it's lovely that you can culture it from an experimental perspective. You can add the Phytophthora cinnamoni containing rice grains to a pot, and then you can add sterile rice grains to similar control pots. And then you can ask the plants, okay, how do you respond to this pathogen? How quickly do you die? What are the disease symptoms like? So experimentally, it's a really nice method. And then we've combined that with the ecological technique of taking whole soil microbial communities and asking about their emergent properties. We've done that for species that are growing at Holden Arboretum, which for us here in Northeastern Ohio, it's right down the street from us. And they have an incredible living collection of plants, rhododendron plants that have been growing for many years in the same location at Holden Arboretum. And the result is that these plants have been conditioning the soil. They've been influencing the soil microbial community at their root zone. And we had some wonderful undergraduates here, Nick and Evan, who are collecting soil right under this particular rhododendron plant. So we took replicate collections per species. We did this for 14 species, and then we did a manipulative experiment with and without the pathogen. So this is with the pathogen and without the pathogen, and then with and without that whole soil microbial community. So as you would predict from the literature, we predicted that live soil biota might have a benefit for plant health. Plants might grow better or survive better in the presence of live soil biota. Certainly, we would predict that the pathogen in general would have negative effects on the plant, but we also predicted that the soil microbial community might be protective and might enhance plant health in the presence of disease. So this was the factorial experiment. Again, we did it across 14 species, and that prediction held in general. So what I'm going to show you is data that is consistent with the prediction that the soil biota was generally beneficial, at least in terms of survival. So here on the y-axis, you see survival proportion. This is going to be an average across the 14 species. It goes from around 30% down here of the plants surviving up to 100% survival. And this is the live soil biota treatment. So with biota like bacteria and fungi, everybody all together in that whole community. And then this is the sterilized control. And I'm going to show you first in the absence of the pathogen. In the absence of the pathogen for the greenhouse experiment, in general, everybody survives. It's a greenhouse experiment, so you're probably not surprised by that. We treated them very well, and they survived across the board. And again, unsurprisingly, if you add the pathogen, survival declines. So you'll notice in the sterile environment, about 60% of the plants are surviving in that sterilized control soil. But we can recover up to almost 80% survival if we add back in a live soil microbiome. So that emergent property of the whole soil microbial community is essentially to protect plants against disease-induced mortality. Soil biota are beneficial, as you might predict from, for example, take-all disease in wheat. So you might have predicted this, at least based on some agricultural data. So this is, we think, very intriguing. This is the average across the 14 species. So what happens for individual plant species? Well, the same pattern holds across species that are susceptible to the disease. So for example, Rhododendron maximum here in the Ponticum group. This is, again, that survival proportion. And when you add disease in a sterile soil, only 20% of the plants are surviving. But if you add back in that live soil microbial community, about 70% of the plants survive. So that is a tremendous recovery of survival simply from adding back in the whole soil microbial community. That is a really strong benefit. That is not the case, though, for species that are resistant to the pathogen. Rhododendron hyperetherum, for example, is famous for its resistance to Phytophthora cinnamomi and is used in breeding programs that breed for resistant cultivars. And you see data consistent with that here. It has very high survival, even in the presence of the pathogen. And there's no significant soil biota effect here. Basically, everybody survives very well with or without soil biota. So how general was this pattern across the group? The short answer is very general. So every time we saw a significant soil effect, it was in that same direction. So I've highlighted those 7 species with an asterisk here across a phylogeny of those 14 sampled rhododendrons. A phylogeny shows us the relatedness among species where closer relatives are closer to one another on these branches. So for example, the deciduous azaleas, the Phytophthora, are a clade, or a group of close relatives. And you see within the deciduous azaleas 2 species that responded significantly to the soil treatment, and both of them benefited from the soil biota effect when there was a disease, when there was a pathogen present. The same thing is true in the evergreen ponticums. The same thing is true in the rhododendrons. And then both of our sampled semi-evergreen tsutsutis were not very susceptible to disease. So we saw very little mortality there. But in general, across all of these groups, you're seeing when they are susceptible to disease, soil biota are beneficial every time. So that's super cool. And as kind of a memory device, I like to refer to this as yogurt for your plants. Now, this is a limited analogy, and I want to acknowledge that right up front. You could think to some degree of the plant soil zone as being a little bit like the gut of an animal. It is very important for health. And there's really solid evidence in animal models that the gut microbiome is hugely influential for animal health. And the purveyors of probiotics, like people who sell you yogurt and kombucha, certainly like to trade on this to tell us we should be eating probiotics to enhance health outcomes. And that seems like a great idea. If you could translate that for plant systems, if you could provide plants with a probiotic product that enhanced their plant health and maybe even enhanced protection against disease and mortality from disease, that would be tremendous. Now, we are admittedly a long way from that, but certainly the bacteria, the fungi, and the oomycete community as a whole can be beneficial. It is not universally beneficial. And we expect that the costs and benefits are likely to depend on conditions. Certainly we see that the condition of whether or not a pathogen is present matters a great deal, but we also expect that things like temperature and moisture will matter. So we are at the kind of beginning of starting to answer this question about whether yogurt for your plants can be developed. But it is a nice way to kind of remember this result. And what in the soil might lead to these effects is basically unknown, but there are a number of hypotheses that emerge from the literature that are worth considering. For example, trichoderma and streptomyces have both been shown in rhododendron systems to be beneficial. There is also some evidence that mycorrhizal fungi in some cases can be protective against disease, including some petri dish studies for ericoid mycorrhizal fungi and rhododendrons. So we expect that mycorrhizal fungi could be part of this, but again, the general mechanisms are unknown. So I want to return to this theme that rhododendrons are an excellent study system for addressing general hypotheses, and just give you a small taste of that for a classic ecological hypothesis. Phyllis Coley developed the hypothesis, the resource availability hypothesis, in 1985, and she suggested that high resource availability environments should select for plants that are fast growers. This idea is fairly intuitive. If there's lots of light and nutrients available, then there should be selection for fast growth to respond to those available resources. If a plant is damaged by herbivores under those conditions, because the plant is growing rapidly and has lots of resources available to replace lost tissue, there should be fairly weak selection for defensive compounds. So there should be low allocation to defense. Alternatively, in a low resource availability environment, you can imagine there's very strong selection pressure for growing slowly. The result is slow growth because there are fewer resources available, but tissues are also relatively more expensive to replace. So if a plant experiences the same absolute amount of herbivory under low resource conditions, there should be fairly strong selection for allocation to defense, because each unit of tissue is expensive to replace, relatively expensive to replace, because again, resources are limiting. So you would expect under a low resource availability environment, high allocation to defense. And you can think of sort of potential poster children for these extremes as like your deciduous azaleas are fast growing and potentially might have low allocation to defense, whereas alternatively you might predict that some of our slow growers like Rhododendron hyperethrum might have high allocation to defense. So this allows us to make some nice predictions, and underlying this hypothesis is the idea of a growth defense trade-off, a very classic idea in ecology and evolution. And we can begin to get at that idea with functional trait studies. For example, we have measured specific root length, and by we mostly, I mean my graduate student Grant, who will tell us about more of his work in a little bit. But his research, he has measured specific root length. High specific root length means a large allocation to length relative to dry weight. So a relatively thinner root, if you will, which is sort of cartooned here. And then a low specific root length is relatively less length per unit dry weight. And you would project that these species have slower growth, at least in terms of their roots. And if there is a growth defense trade-off, then you would predict that the specific root length trait would correlate with disease susceptibility, like susceptibility to Phytophthora cinnamomi. And so you predict a positive correlation between these two things, because plants that grow fast and have more root surface area should be more disease susceptibility, whereas slow growing plants should be selected for more disease resistance. That is, they should have lower disease susceptibility. So you expect a positive correlation between these two variables if there is a growth defense trade-off. And what we find in our initial forays into rhododendron research is essentially consistent with that pattern. It's especially consistent in the live soil treatment. So you see it's statistically significant here in live soil, where again, there's a positive correlation between specific root length and susceptibility to Phytophthora cinnamomi. So each of these points is a single species in a multi-species phylogenetic regression. So the general trend holds and is consistent with a growth defense trade-off. Species sort of down at the lower end of this have a low specific root length and potentially are allocating more to defense, or at least are less susceptible to disease, which is consistent with less allocation to defense. So what we see in general, like I said, there are some species clearly that follow this trend. Rhododendron atlanticum, for example, is our species with the lowest specific root length. And it also has amongst the lowest disease susceptibility. It survives incredibly well in experiments. And alternatively, Rhododendron minus is amongst our most disease susceptible of the 14 species we sampled, and it has a very high specific root length, consistent with that trade-off hypothesis. But I do want to point out that not every species sort of perfectly fits this projection or this prediction. For example, Rhododendron hyperethrum has very high specific root length, or at least did in our experiment, and also had very low disease susceptibility, suggesting that specific root length, unsurprisingly, is not capturing all of that trade-off allocation space. So certainly a great deal more work needs to be done to fully understand growth defense trade-offs and their extent and limits across rhododendron. But this is a really excellent study system for starting to address those questions. And I just want to return again to that theme that I've been telling you about all along, which is that there is replication of disease susceptibility within multiple groups, within multiple clades of rhododendron. So this is why I, as like a scientist who kind of likes phylogenies, gets really geeked out about rhododendrons because they are incredibly powerful. So you see here the deciduous azaleas. I've marked them in salmon. They're a clade, and they contain both highly susceptible species. I've marked the more susceptible ones in bright pink, and less susceptible ones. So the species that have a white basically had no mortality when we added the pathogen, whereas the ones with the darkest pink had the most mortality. And you see there's a tremendous amount, this is a quantitative scale, so there's a tremendous amount of quantitative variation across species, but even cooler, there is variation within groups. So within Phytophthora, there's a lot of variation, some very susceptible species and others that are very resistant to disease, or at least that aren't dying in our experiment. And again, that's true like down here in rhododendron, one of the most susceptible species in our experiment, Rhododendron minus. Its close relative, Rhododendron kieskii, very few of them died experimentally. And so this replication across groups allows us to ask interesting questions like, not only is there sort of a benefit of the soil biotic community, not only is there the possibility of yogurt for your plants, but if yogurt for your plants were something one could develop, it might be general across groups. It might not just be an idiosyncratic special thing that only deciduous azaleas can respond to. In fact, it might be a general thing. And as scientists, we would certainly like to understand where are patterns general. In addition to getting sort of geeked out about just like the beautifulness of the phylogenetic results, this is also just a beautiful group aesthetically. There's a reason people love looking at them, love putting them in their yard, and love them as hobbyists. So I think they are really ripe for doing community science. And we're piloting a very small, at least initially, very small community data based project. We're calling it RA-LEAFY for Rhododendron and Azalea leaf traits. And what do I mean by community science? Well, we're modeling it after other community science projects like Project Budburst, which is run by the Chicago Botanic Garden. And what they do is they ask people to measure when do the leaves burst open on plants in your region. So you can imagine doing that in your backyard. You could either take your smartphone and upload data to their app, or you can upload data to their website later. But as long as you can identify the plant, you can upload data about when the leaves burst open, which tells us a tremendous amount about how plants are responding to climate, including shifting climates over time. So community science is really powerful because if you can tap into something that's easy to measure, and that's very important, that's easy to measure, that you can do ideally on an app, on a smartphone, or on a website, it can be something that you could get people all around the world doing. And I want to give you just a tiny flavor of what we hope to do with that. I've had a wonderful undergraduate working with me this summer who has gone out to Holden Arboretum and looked at rhododendron plants, and he's measured their leaves, and he has measured leaf damage. And just to give you a small flavor of that, for Rhododendron atlanticum, what he observed is almost no leaf damage in the field. And that's very similar to Grant's greenhouse experiments, where he experimentally inoculated with Phytophthora. So this is very anecdotal, of course, but we see very high disease resistance in this species in the greenhouse and almost no leaf damage in the field. We have no idea what mechanisms might cause leaf damage in the field, but again, it's super easy to measure. So that's awesome. Also, similarly, Rhododendron brachycarpum had very high mortality in the experiments, and NOAA in his observational work observed a lot of leaf damage. And NOAA can take these images. You can imagine that once we've developed an app, you can take an image with your smartphone, or you could upload them to a website, and NOAA can digitize these images and quantify the percent of leaf that is damaged. So in general, we're going to be able to use this over the long term to address broad ecological and evolutionary questions like that about whether there is a trade-off between growth and defense. And again, this is just barely being piloted, but I would like to encourage you, if it sounds interesting to you and you'd like to know more or be in touch, please consider emailing me and just asking me questions about it, or I'd love to be in contact with you. And I think that this presentation and my email address will be available to you later, so you can always freeze the video again and get my email address that way. I'd love to be in touch. So I'd just like to emphasize these final points. We found that the soil biotic community could be beneficial, at least in the context of survival. And Grant will tell you more in his presentation about some of the sort of alternative findings that we're finding with biomass. And that suggests that there is the possibility, at least, of developing probiotics for plants for the soil. And that could be an incredible future benefit as climates shift and food security becomes more and more challenging. And I just want to re-emphasize again, finally, this very interesting pattern that we find, that disease susceptibility varies a lot amongst these rhododendron species, suggesting that they are a powerful study system for addressing these general questions as we are continually challenged by the pressing problems of climate change, disease, their potential interactions, and so forth. So we're just beginning to get into those questions now. And I want to say, before I wrap up, thank you to all the people and funding agencies who made the work possible, especially our wonderful collaborators at Holden Arboretum, like Juliana Medeiros, your host today, Steve Krebs, who taught us how to do hand pollination of rhododendron and how to do the rice inoculation, Francesca Hand at Ohio State University, who's provided more Phytophthora cultures, many wonderful undergraduates like Nick, Alexis, and Evan, and Grant, who did almost all of the data collection that you saw today, many other undergraduates, including Noah Clayton, who is helping to pilot that leaf data, and the American Rhododendron Society for funding the experimental work, the Expanding Horizons Initiative as well at Case Western Reserve University for funding the pilot of the RA-LEAFY database. And if I have a little bit of time, I would love to try to address some of your questions. Thank you all so much.
Thank you, Jean. Let's have a nice round of applause, virtual applause for Jean, if you can show your little clap emojis. Now, we didn't get any questions in the chat. Oh, except Bob Weissman does have one. Bob says, thanks for your great talk. It's my understanding that sudden oak death is caused by Phytophthora ramorum and not cinnamomi.
Oh, thank you. You know, there are probably multiple causes of sudden oak death, and that's a very good point. The experiment or the observational study and model that I was telling you about was for Phytophthora cinnamomi, I believe, but you're probably right that more sudden oak death is caused by Phytophthora ramorum. As a non-Phytophthora expert, thank you. That's very helpful.
I had a question for you, and anybody else who has a question, we do have a few minutes, so please put your questions in the chat. I'll just go. One of the things that I think about is this idea that people have often said, well, there's definitely pathogens in the soil, right? So there's Phytophthora. Yeah. So how do you know where to get good soil? When you guys do your inoculations, do you look for healthy plants? Like, how do we know to get good soil that's going to be good for the plants and that we're not getting pathogenic soil instead?
Right. So, I mean, for our experiment, we did not specifically target healthy plants. And in fact, we tried to target plants haphazardly, not strictly randomly, because we were somewhat limited by what was available. But we tried to target plants certainly that were alive and available to us, which to some degree does mean healthy, right? Like at the Arboretum, especially if they see strong signs of root rot, they are going to yank some of those plants out. So we probably have kind of an inherent bias toward healthier plants because we're sampling at an Arboretum. Now, I'm not necessarily saying to people, oh, in your home garden, you should do this. You should, put this yogurt on your plants right now because it's going to improve their health. Because I think we're still at the beginning stages of understanding all of that variation in the soil microbial community and what its complicated effects are. First of all, we don't know that we didn't sample pathogens when we sampled that soil. We probably did sample pathogens. And it also could be the case, interestingly, that if you sample a community that has quite a few pathogens, but the plants are fairly resistant to those pathogens, you could be inoculating the plants with a community of pathogens and essentially inducing systemic resistance, which leads them to be a little more resistant to Phytophthora infection. So we could absolutely be inoculating not just with mutualists, but with pathogens, and unexpectedly or unintendedly having a positive effect on survival as a result. So I think a lot of the ecology is yet to be worked out about like what's going on in that community of soil microbes. I think there's so much to learn there. I know I loved the analogy yogurt for your plants because it's so easy to remember, but of course I want to acknowledge that it has so many limits.
Yeah, whenever you're talking to people who are new to something, it's always good to have an easy, handy analogy, but then it starts to break down the more people.
Analogies
so
always
we'll just love it.
But
we'll love it for what it is. We don't need
right.
it to be exact. I've got a couple more questions for you here. Let me read them.
Oh, I can see the chat now, and I can see a question about commercially available mycorrhizal fungal powders. Certainly there are some. And I am not dismissing them as snake oil, nor am I saying that they're always universally beneficial. Like we've done some experimental work in other contexts, like in restoration context with commercial products, and that work in our lab has been very limited. But we've found that the effects of the mycorrhizal inoculum were surprising and unexpected, I guess you could say. So we added in a field experiment with, this is not a rhododendron experiment, but a tree experiment. We added a mycorrhizal inoculation. We recovered when we did high throughput sequencing, no mycorrhizal inoculum, like none of the things we inoculated were recovered, but we did recover some unexpected pathogens. We also recovered some mutualists that weren't in the inoculum. And so it can be really hard to know in a broad field context how much inoculum you need and when it's going to be beneficial or when it's not. So I can't tell you, is it the best thing since sliced bread or is it snake oil? Yes and no for both. It totally depends on the environment. And I still think we're getting our hands around that as a field. Like we don't know the answer yet. That's a great question.
Yeah. So there's another question from Connor Ryan. How does growth defense trade-off mesh with tolerance? My experience with Rhododendron ponticum is that despite being Phytophthora susceptible, it seems to perform well under mild disease pressure because it can just produce new roots at a rapid rate. Plausible?
Yes, I love that question. That's so cool. So yeah, the idea of a growth defense trade-off, right? It's obviously oversimplified. Like it's a cool idea and we sort of love it in ecology and evolution because trade-offs make so much sense. But if you grow quickly, I think you probably can tolerate a certain amount of pathogen pressure. If you lose a little bit of roots to the pathogen, you can still recover by producing new roots. That absolutely makes sense to me. I think it's very plausible. And I think it's part of that kind of overall like trait tolerance resistance space, which is a multidimensional space. So I think as we move forward in the system, we're going to get more and more multidimensional, and we're going to start thinking about not just resistance per se, but resistance, tolerance, and how sort of multi-trait trade-offs and synergies play out in that space. So I think it's going to get much more kind of nuanced and interesting, of course, the further into it we get.
Jean, could that explain the results with hyperethrum? So you had that hyperethrum having really high specific root length. So if your roots were really cheap, you could just chuck them off at the moment they got an infection and grow a new one. Could that be a plausible explanation for that finding?
I think to some degree it is plausible. I think it probably doesn't explain all of the results because rhododendron minus has a very similar specific root length. So a specific root length were the trait that totally captures that like root turnover strategy, which probably it doesn't, but if it captured that root turnover strategy, then we would expect the same pattern for minus and for hyperethrum, but we don't see it. So I think it's got to be more than just, it's something that's more than captured by specific root length.
Maybe it needs an immune response. So if you don't have the immune response to chuck the infected root and you just keep growing gloriously unawares.
Makes so much sense. Yeah.
Very
absolutely.
cool. All right. I don't want to get, Jean,
how much time do I have to answer questions?
We've got a couple more. We've got about 5 more minutes if you want to keep going.
Okay, so there's one more question, at least in the chat, which was the live soil collected from under the species to which it was applied? Yes, I'm so sorry. I forgot to say that. Yes, it was. So for each of those 14 species, we went to the conspecific, so to that same species, collected 3 replicate collections under that specific species and inoculated that species with its own conspecific soil microbial community in the greenhouse. We did that in part because we thought that microbial community might contain species-specific mutualists, but also just sort of from a feasibility standpoint, you can imagine an experiment where you do try to look at heterospecific conditioned soils, and ecologists love those experiments because they're very powerful for asking very interesting questions about coexistence. But with 14 species and 13 heterospecifics, the experiment starts to get pretty not feasible pretty quickly. And I think my wonderful team of graduate and undergraduate students would probably have murdered me if I had said we had to do that experiment. So we definitely thought about the heterospecific treatment. We haven't done it yet. But I think there's so many cool questions you could answer once you start to go that direction too.
You had about 1,400 pots in that experiment, or how many pots did you have? You had a lot of pots.
We had almost 3,000 individual plants, and I don't remember how many individual pots. Grant, do you want to chime in? Do you remember the pot number?
Yes, there are 672 pots.
672 pots.
Right.
So doing the factorial with the heterospecific soil would have multiplied that by 100, a lot.
Yeah, and he and to some degree the team as well, but he measured the root traits of all of the individuals. There were 3 replicate plants per pot. So almost 3,000 plants. He spent all of the fall semester measuring root traits, all of it. So, and that's while I was on maternity leave. So, you know, imagine the feasibility, I guess, is part of the answer to that question about conspecific versus heterospecific soils. We think the conspecific soils is super cool, but I would also love to do a heterospecific soil experiment at some future date. I think that'd be interesting.
Or maybe even a different clade treatment.
Or like a within clade versus among clade. I mean, there's
susceptible versus not.
Co-occurring in the field versus not co-occurring in the field.
So I guess what we're saying is you've got plenty of work left to do.
I'm saying, like, you know, if you want to be a grad student or an undergrad in the lab, you know, come do a master's, come do a PhD. So many cool things you could do.
Wonderful. Well, thank you very much. This was a fabulous talk. I think everybody you've got a lot of thank yous in there.
Great questions, super fun. Thank you.